Heterogeneous clinical course over time. Several prognostic markers have been developed for CLL, including lymphocyte doubling time, disease stage, serum markers, CD38, ZAP70, mutational status of the immunoglobulin genes, and cytogenetic aberrations. However, other molecular biomarkers are yet to be discovered. Of these markers that might contributing to the CLL activity and the prognosis is the programmed cell death protein 1 or PT1. Without doubt, T cell lymphocytes are the main effector anti-tumor cells. T cells recognize potentially antigenic peptides from either pathogens or tumor cells presented to them through antigen-presenting cells by major histocompatibility complexes to be recognized by the T cell receptors. In addition to this presentation from the antigen-presenting cells, T cells require further interactions, which is known as co-stimulation. The PD-1 is a co-stimulatory cell membrane protein receptor from the immunoglobulin family. It acts through interaction with its ligands PD-L1 and PD-L2. These interactions between PD-1 and its ligand inhibit effector T cell function, regulating injurious immune responses and blocking the autoimmune process. Normally, PD-1 is expressed in hematopoietic cells, including lymphocytes, myeloid cells, macrophages, mast cells, as well as non-hematopoietic cells, including epithelial and endothelial cells. Its ligand, the PD-L1, is the main receptor ligand on several tumor cells, like solid tumors. It's very well known in solid tumors. However, it was found to be expressed in hematological malignancies, including CLL, acute leukemia, and multiple myeloma. CLL is characterized by immune suppression manifested early in the disease, and CD8 positive T cells in CLL express the PD-1, and this could anticipate the immunosuppressive tumor microenvironment. However, malignant CLL cells also overexpress the PD-L1. This dysregulated PD-1-PD-L1 interaction contributing to the strong inhibition of anti-cancer T cell responses and resulting in disease progression. Epstein-Barr virus is a human-specific gamma herpes virus that infects B lymphocytes. It typically affects more than 90% of the population worldwide. And the EBV is characterized by its latency in the B cells, and it has the ability to transform the normal B cells into tumor cells. In spite of not being an EBV-associated disease, CLL B cells or B lymphocytes, malignant B lymphocytes, have been shown to express the Epstein-Barr virus proteins receptors on their surface. Furthermore, there is compelling evidence that Epstein-Barr virus infection predisposed to different B cell tumors, including endemic Burkitt cell lymphoma, HIV-related lymphomas, post-transplant lymphoproliferative disorders, aggressive lymphomas of the elderly, and lately CLL. As we previously mentioned, that antigen recognition, such as in chronic viral infection, might lead to T cell exhaustion and suppression, resulting in detrimental immune reaction to cancer cells. Therefore, Epstein-Barr virus infection might predispose to the same process. Several recent studies proved that B cell lymphomas that are associated with Epstein-Barr virus infection express the PD-L1 that binds to the PD-1 on T cells, inhibiting its toxicity. In the same context, we thought that CLL might show dysregulated expression of both PD-1 and its ligand in PD-L1 in CLL. That's why in this work, we aim to study the expression profiles of PD-1, PD-L1, as well as Epstein-Barr virus viremia in patients suffering from CLL and whether they affect CLL prognosis or not.
بروجرام سكرين لو اشتغلت الفيو كده كويس مريح ولا اعمل فيو تاني؟
لا تمام، لا مش هيفرق. The current work was carried out in 80 CLL patients admitted to Alexandria Main University Hospital in the period from 2016 to 2019. They were newly diagnosed according to the WHO criteria, treatment naive, aged from 45 to 73 years old. 35% of them were females and 65 were males. And we followed up our patients for a median period of 18 to 48 months. CL patients with second malignancy or those who received treatment, received recent blood transfusion, signs of infection less than 2 months before enrollment in the study, or autoimmune diseases were excluded from the study. 80 control healthy subjects, hematologically and cancer-free, aged from 45 to 70 years old, and sex matched to the patient group were considered as a control group. Patients were modified according to the modified race staging system. CD38 and ZAP70 expression were studied on peripheral blood viable lymphocytes by flow cytometry. Fish analysis was carried out using cytocell CLL profiler kit, which analyzed the basic 4 chromosomal aberrations in CLL. 40% of our patients showed 13q14 deletion, 23% showed ATM deletion, and 15% showed p53 deletion, and 18% had trisomy 12. Regarding Epstein-Barr virus analysis using real-time PCR, DNA was isolated from peripheral blood nuclear cells. Quantification of the Epstein-Barr virus DNA load was assessed by real-time quantitative PCR using Reuters real-time PCR system. PD1 and PDL1 expression analysis was carried out by RNA extraction from peripheral blood nuclear cells according to the manufacture and structure, followed by assessment of concentration and lethality by concentration by nanodrop, reverse transcription by the high-capacity cDNA transcription kit, and finally the PD1 and PDL1 expression profiling by real-time PCR or strategy real-time PCR system using cyber-green Fiagem master mix and sequence-specific primers. Our results showed that 47% of CLL patients were positive for Epstein-Barr virus infection, and 33% were negative, while in the control group, 37% were Epstein-Barr virus positive, and 62.5% were negative. The EPV DNA load in the study participant was significantly higher in the CLL patients compared to the controls. PD1 and PDL1 expression profile was significantly higher in CLL patients compared to the control groups. Patients were categorized according to the EPV status into CLL patients with positive EPV and negative EPV. Patients with positive Epstein-Barr virus infection showed significant association with ZAP70 positivity, CD38 positivity, and higher categories of the modified race staging system. Regarding cytogenetic aberrations, there was a statistically significant association between Epstein-Barr virus positivity and ATM deletion. Regarding the clinical outcome, Epstein-Barr virus positive CLL patients showed higher significant need to treatment and was associated with poor outcome regarding the achievement of complete remission or the death rate. PD1 and PDL1 were significantly higher in Epstein-Barr positive CLL patients compared to negative ones. There was a statistically significant positive correlation between the expression levels of PD1 and PDL1 and the Epstein-Barr virus load in CLL patients. Patients were further classified into, according to PD1 and PDL1 expression, into those with high expression and with low expression based on the median expression value of both markers in each patient. Okay. Both markers, high expression levels were significantly associated with positivity for ZAP70CD38 and higher stages in the modified Rayleigh stage, and significant association was found between PD1 high expression and the trisomy 12 and the p53 deletion, while PDL1 was significantly associated with the p53 deletion. High expression of both markers was associated with higher requirement for treatment, failure to achieve complete remission, and higher death rate. Both markers, PD1 and PDL1, was significantly positively correlated together in CLL patient as a whole and in Epstein-Barr virus negative and positive subcategories. We used the log-rank test and the Kaplan-Meier curves analysis to determine the target gene expression profile relation with the survival, and we found that a high expression of PD1 and PDL1 was significantly associated with a shorter survival in CLL patients. Similar results were found regarding progression-free survival. Multivariate analysis revealed that ZAP70 was considered as independent predictor for shorter progression-free survival in CLL patients, and both the p53 deletion and the PDL1, not PD1, were independent predictors for shorter overall survival in CLL patients. We concluded that PD1 and PDL1 are upregulated in CLL patients. Our study suggested that Epstein-Barr virus infection affects CLL prognosis negatively, and this might be due to high PD1 and PDL1 expressions. Furthermore, Epstein-Barr virus infection in CLL patients is linked to increased risk of treatment failure and poor outcome. PDL1 high expression level was found to be independent predictor for overall survival in CLL patients. And in addition, PD1 and PDL1 might be possible therapeutic targets for patients with aggressive CLL based on the fact that they are effective targets for immunotherapy in other cancers. This research work was published in Clinical and Translational Oncology, and I want to thank the rest of the team with me, Dr. Omar Ghalab, Dr. Marwa Gamal Eddin, and Dr. Iman Atiyah, and finally thank